Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. In this content, we're going to be investigating how phospholipase D, an enzyme in muscle, impacts muscle hypertrophy or muscle growth.
Learn Your Body, a science-based education.
Now, this particular research comes from some data that came out of a paper that I fully dissected, and I've got that paper linked to this content as well as my notes and any amendments that might follow this content's publication. So with that said, let's jump into a little bit of the methods. It's pretty simple. It's very mechanistic, but it's still quite simple in how they went about this study. So how did they go about it? Well, they looked primarily at cells, so they wanted to look at myotubes, which are what are essentially considered differentiated muscle cells, meaning that you have a premature muscle cell known as a myoblast, and then you can convince it essentially to turn into a myocyte or a myotube in this particular situation. So we're looking at muscle cells that are just plated on a dish. And then there's a little bit of data in mice as well, looking at this phospholipase D pathway, but the primary parts of this paper are all in cell culture. Now, briefly, what is phospholipase D? Phospholipase D is an enzyme that's found on the sarcolemma of the muscle, meaning the cell membrane of the muscle. So it's right on the edge there, right at the top, right where you have this protection from the outside of the cell to the inside of the cell. It's right on the inside of the cell. And it actually produces a particular molecule known as phosphatidic acid. Now, phosphatidic acid, I'm not going to go into all the different pathways right now, but the production of phosphatidic acid has an impact on the master regulatory unit of cells for protein synthesis and growth. That is, you may be aware of it already, mTOR, or mammalian target of rapamycin, also known as mechanistic target of rapamycin. There's a bunch of different names, but it's mTOR. And if that is increased in its activity, then typically you see increases in protein synthesis and cell growth. So that's how PLD, phospholipase D, impacts the protein synthetic pathway, or at least that's what we're hopefully going to try and figure out. So now in this first piece of data, they are essentially adding two different drugs to cells. Again, these myocytes or myotubes that are just on a plate, and then they just want to investigate what they can do to these cells. And in this situation, what they're doing is they're adding two different drugs in different situations. So in one situation on one plate, they're adding 1-butanol, which impacts phospholipase D by inhibiting phospholipase D specifically. Then they've got a very similar molecule called 2-butanol. Now, the reason why they want to use a secondary molecule there is because it does not affect phospholipase D, but because it's so similar, then it gives more credence and more specificity to that first treatment of 1-butanol. Because then you know that there's some specificity with 1-butanol, but not only that, you also know that similar molecules won't have any sort of off-target effect. So there's a number of different reasons why you would want to do this, and it's essentially what's called a control in an experiment. And it's pretty important to have, otherwise you can't interpret the actual results of the study. And you can see by the images here of the actual cells, they're kind of elongated, that with 1-butanol you see decreases in the muscle cell size, but in the other two images you don't see any sort of decreasing of the cell size. Now granted, I will say that in this image they do a pretty poor job of actually showing this. This is not a great quality image. I wish that they had improved this substantially, but they do have the quantification in terms of bar graphs, so you can see that there's a dampened effect from the inhibition of PLD in this situation. Now in the next set of experiments, they're doing a similar thing, but they're adding something to this experiment that's similar to what we saw with the butanol treatment. This is using an siRNA, or silencing RNA. And essentially what a silencing RNA does is it essentially inhibits a particular gene, and in this situation we're inhibiting, as you can guess, most likely phospholipase D1, which is the one that we're most focused on, while the other one that they also test is phospholipase D2. Now that one should not have as significant of an impact on muscle hypertrophy. But phospholipase D1 is the main one that we're focused on, and that one does seem to have an impact on muscle hypertrophy, as we'll see. So silencing phospholipase D1 does decrease the cell size. However, silencing PLD2 does not seem to have an impact, as you can see by comparison to the control treatment, which is the addition of some scrambled RNA, meaning an RNA that's not going to silence any gene that we're interested in. And then another treatment that they wanted to add in there was what would happen if you increased the expression of PLD1. Do you see increases in hypertrophy? So we've knocked down PLD1 and we see that that decreases the size of the muscle cells, but can we increase the size of the muscle cells? So what they do is they use what's called a transfecting agent or transduction agent, however you want to end up saying it. It's essentially a virus that encodes for a particular piece of DNA, and in this situation that DNA is PLD1. So they inject this virus into the cells and it integrates into the cellular structure, the DNA structure, and then you get more expression of that PLD1 gene. So with increased expression of PLD1, what do you find? Well, interestingly enough, you see increases in muscle hypertrophy. So with greater PLD1, there is greater muscle cell growth. Now, a little bit in mice, they did the exact same experiment with the adenovirus, which is that virus that I was telling you about in the previous experiment, but they're injecting it into the muscle of a mouse, and they want to see if they overexpress PLD1, what's going to happen. And sure enough, it actually happens in an in vivo system, meaning that it happens in a live system like a mouse. You do see increases in the total muscle belly size. So it's not just the single cells, but you're actually seeing it in an in vivo system, in a physiological system. And the final experiment is to look how the impact, if they add a glucocorticoid, which is an atrophy stimulating molecule or hormone that would typically decrease the size of the muscle cells, and it does in the condition that they only use the glucocorticoid. But then if they overexpress PLD1, suddenly you see a rebound of the size of the muscle cells. So the PLD1 seems to protect against the atrophic conditions that are presented with the addition of glucocorticoids. And then another fascinating aspect is that they bypassed PLD1 and they just added phosphatidic acid, which is the product of phospholipase D, which is what I was talking about earlier. So if they bypass PLD1 and they just straight up add phosphatidic acid, they also see a rebound in the muscle growth potential of the muscle cells, even in a glucocorticoid environment. And the final piece of data is looking how PLD1 overexpression, or the expression or the inhibition of PLD1, how that impacts atrophic genes, so atrophy being the shrinking of the musculature. And what you find is that when you overexpress or when you have greater activation of PLD1, then you see decreases in these atrophic genes, so the 4 that they measured here. Now, they're measuring gene expression, they're not necessarily measuring the proteins themselves, which would probably be preferable in a situation like this, but I think it's still a good proxy of information based off of the information that we already have from the glucocorticoids. So here they're giving some further proof that PLD1 not only stimulates potentially hypertrophy, but actually has a braking mechanism on the atrophic side, so the degradation side of muscle loss or muscle maintenance or muscle growth in general. And actually, I lied. That was not the final piece of data. I do want to include this last piece as well. So they looked at atrophic genes, but they also want to look at mTOR, if PLD1 runs through an mTOR dependent pathway, and it seems that it does, because when you inhibit mTOR and overexpress PLD1, you do not see increases in muscle cell size. However, when you leave mTOR perfectly normally expressed, and you overexpress PLD1, it does seem to have an increase in muscle cell size. So it seems that based off of this evidence that PLD1 does run through an mTOR dependent pathway, although there are mTOR independent pathways for protein synthesis and cell growth in general as well. It's just that mTOR is known as, again, like I mentioned earlier, the master protein regulator. So what can we take away from this paper? Well, that phospholipase D does decrease muscle atrophy while increasing muscle synthesis or muscle growth. And it does that with the production of phosphatidic acid, and that it is mTOR dependent. So if you don't have mTOR or you inhibit mTOR, then you will not see that effect through phospholipase D and phosphatidic acid. So hopefully you found this informative. Hopefully you thought this was kind of cool. In terms of how we can actually use this information, that's going to be tough to do because you'd have to find some way of overexpressing phospholipase D, or if you want to bypass it, maybe sometime there might be some supplement for phosphatidic acid if there isn't one already. But in terms of delivery, that's a whole nother thing. So that's a discussion for another piece of content. But hopefully you found this informative. And with that said, I wish you a wonderful day, and I hope I have the pleasure of speaking with you in the next one. Have a good one, guys. Bye.